fine. The second thing which we will going to study is about the tumor suppressor genes, the brakes of the cells. As I told you, they are like a more or less like the brakes. They are the stoppers, controllers, and down regulating the cell cycle. And it's very important for the cells to have these things because they will let the cells know that it is time to go, time to stop the cell division. And remember, many human cancers are because of the loss of functions of the tumor suppressor genes. Think about the proto-oncogenes. If they gain their function, it can lead to cancer. Similarly, tumor suppressor genes, if they lost their function, it can contribute in cancers. So there are two types of tumor suppressor genes are out there. One are called as gatekeeper tumor suppressor genes. The other are called as caretaker tumor suppressor genes. The gatekeepers are the ones, they came from the genes encodes and they produce a proteins which either regulate the transition of cells through the checkpoints. So they are regulators, gatekeepers. They check and see whether the cell should pass the G1S and then the cell should pass the G2M. They will say yes to these type of things and then the cell will go for the cell division. They are the gatekeepers of the cell cycle. All of them, the big things which you have, which you can read in the earlier slides of cell cycle, most of them are the gatekeeper tumor suppressors. There are other type of tumor suppressors which are called as caretaker, which take the job, which take care of the functions going on within the cells. They are the proteins which involve and which can detect and repair the DNA mutations. So the normal chromosomes and the integrity of the chromosome survives within our genome. It's very important that the human genome we are carrying is as a result of like evolution of millions, billions of years. So nature has created a DNA repair mechanism as well along with this to keep the integrity of that genomic DNA. So anything which actually affects the sequence of that DNA is dealt with very strong hands. And tumor suppressors are the one, they can detect the error, they can repair the mutations as well, and they help the chromosomes to stay in their normal with their normal integrity, with their normal sequences. And nothing should nothing should harm the chromosomal structures and integrity, even during the cell divisions as well. They are the brakes of the cells. Whatever we were studying earlier, all those were accelerators. All those were the genes which push the cells to go for cell division. And whatever we are studying, those are the genes which help the cells to slow down, not to go for the cell division. And they are of two types. The one which check the cells that everything is fine, which helps the cells to pass into the G1S and then G2M, checkpoint regulator. And then they are DNA integrity regulators. So they are different topics. Ji Nimra, aap kuch keh rahe?
Ji sir, you told P21, P27 and P57, they could act as a tumor suppressor, right?
Exactly. देख एक्टिव ओके हम पढ़ेंगे यहाँ पे वो देख आ गए अब बहुत लाइक है list of tumor suppressor genes. Okay, there are some of the gatekeeper genes. RB1 is a very famous one. This one, P53 पूरी दुनिया पढ़ती है. You should think about other examples than P53. Okay, retinoblastoma associated protein RB1. Okay, any error here, it will lead to the cancer called retinoblastoma. Also, you should know that the name of the gene of P53 is TP53. The name of the protein is P53, but the name of the gene is TP53. And once again, all these are symbols are in italics. So all these are cell cycle regulator. I highly suggest you that you should be able to interpret at least half of them. Okay, their job, their function, and how they do their job, how they control, how they regulate, and act as a gatekeeper gene. The other is the caretakers. The BRCA1 is here, the BRCA2 is here. Oh, you can see DNA damage repair pathway. they're not working well, they can also lead to the trouble and cancer. And BRCA is very famously associated with breast cancer, and as well as with the ovarian cancer. Okay, so the next slide is about, oh, we don't have time. How come? How come? You should remember that difference is that if this mutation is required during our lifetime, because it is called somatic mutations. We know these things, no? Somatic mutations, because we are acquiring mutations during our lifetime. So if we develop that mutation on that gene during our lifetime, and it can be passed on to the next generation. We might develop one mutation on the gene on one allele, and accidentally, if we are unfortunate enough, we might develop after developing the first error, we might develop the error on the other allele as well. The
नॉन
function,
हेरिडिटरी के फैक्टर तो सर नॉन हेरिडिटरी के फैक्टर्स क्या हो सकते हैं
like whatever
मतलब एक
the
तो हेरिडिटरी
factors,
मतलब है कि वो पेरेंट्स से आ रही चीज ना
whatever the factors which can cause mutations in the DNA. There may be radiations,
ओके
there
ओके
might be toxins, there might be chemicals, and our food, dietary habits, and stress, whatever which can cause mutations. Okay, but the unfortunate
ओके
situation is that somebody develop a mutation on one allele in his lifetime, and then unfortunately he developed the mutation in the other allele as well, and that is the only way it will going to lose the function of that thing, and that will lead to a single tumor in one eye. But if it is transferred through
ओके
like hereditary means, the error transferred through this means, in that case the tumor will be present in both the eyes. It's quite different, and that is you can differentiate between these two. So let me give another try after Rubab. I should this has explained by Rubab. Let me give somebody else a chance to explain the same things in a different way. Look at that. Is the same thing is here? Who is the brave guy or a lady who wants to explain?
Can I?
The same thing is present on this slide. Eamon has raised the hand. Nimra has raised the hand. Somebody else wants to explain? Who is clapping instead of raising the hand? Somebody. No, Kadesh. I'm a little bit biased. Let me ask Kadesh to answer this. Kadesh, आपकी बारी है. Explain करें. The same things are present on this slide as well. Okay. Okay, please.
Sir, as I was reading about the previous slide, the RB thing, I noticed that E2F thing there. So I searched it, and it was a transcription factor. So after the RB attached to the E2F, then it produces the gene, and which is the normal one. And if that RB is mutated, it would not be able to attach to this E2F, and so sir, could you go to the previous slide?
No, you are explaining well. Actually, I am trying and helping you. Okay.
Okay.
Could you please see this is where the RB and E2F is not bound with each other. That's a problem. Okay.
Yes.
Can you see?
So
it does not bind with each other, and that will lead to the activation of this gene.
Right.
So,
yes, sir.
So the genes. Shabash. You can.
It would be phosphorylated and it would be inactivated. So that's the question from my side to you as well. Like sometimes we read that after getting phosphorylated, it gets activated, and this time it is
okay. Let me answer. You need to see the local situation. What's going on here? Okay, look at me. Activation means for the protein is doing a job. He is not doing the job. That protein is inactivated. If somebody makes that protein to do its job, that stuff and that protein is now called as activated. That's a different thing. Okay, that's a local thing. Now see, RB protein is here, and that RB protein is able to bind with the E2F. It able to bind with the E2F since the RB protein is phosphorylated. Or you can say hypophosphorylated, means it is a kind of less phosphorylated. That creates a conformation which allows the RB protein to bind with the E2F. Once this thing is established, the downstream genes, who are the downstream genes? The downstream genes are the ones which can controls the S phase, If the genes from the S phase are not activated, they're not expressed, you can guess the cell will not move to the S phase. So this combination means the cell is halted at that state. Now think another situation. If the RB is got hyperphosphorylated, it has more phosphorus. In that case, it will not, its conformation is not accurate enough, because the hyperphosphorylation will change the dynamics of that protein, will change the structure of that protein. It will not be able to bind with the E2F. This connection is not possible. So that transcription factor, which is supposed to express the genes of S phase, are now free. So it can proceed and start making the messenger RNA. They will produce the proteins, and these proteins will activate the S phase. So that is how the S phase will be activated. So the concept of activation, expression of genes are all subjective, or all like a matter of a local story. Here you can see more phosphorylation changes the conformation, not allowing the RB protein to bind with E2F. E2F is free and it will going to push the expression of genes. In this case, RB protein with the less phosphorylated version has its shape, which can allow it to bind with the E2F, and it will not going to express any genes. So it really depends what these things are doing here. But you can see RB protein is somebody who is stopping the genes to express. Stopping how? Stopping by binding with the E2F. If somebody changes the RB protein will not be able to bind with the E2F, and that will kept on going, producing lots of proteins, pushing the cells to go for the S phase. Can you see this clear?
Yes, sir. Thank you.
Please do not generalize things in life sciences. I mean, एक word देख, अभी वो जैसे हमजा ने पूछा था. It's not about one single factor, which is you need to know all the picture of the story, and then you need to make your conclusions. Okay? रुबाब इसको annotate करने के बाद delete भी करते होते हैं. Okay, here we go.
सर मैं ट्राई कर रही थी लेकिन वो हुआ नहीं है,
चलो
पता
कोई
नहीं
बात
क्यों नहीं रही.
कोई बात नहीं, forget it. हम बाद में कर लेते.
Okay.
Okay, this is an example which I have explained you earlier, that how the functions of a tumor suppressors and an active oncogene actually works together. So and the regular work, a tumor suppressors works like the brakes, and the proto-oncogenes, they work like an accelerator. An active oncogene can be countered by a tumor suppressor. It can stops or slows it down. But without tumor suppressor, this will lead to an accident. It's not easy. So that is how both of these things are required for the normal function of the cells. And this is an example of retinoblastoma in the eyes. And then one more slide, and then we can conclude. I think it's now clear at this time that if the P53 is available, it will going to express the target genes. So one of the target genes is like P21. And if it is there, it will suppress the cyclins and the CDK and does not allow the cells to go for the cell division. Now, alternatively, if the P53 is not functional because of any XYZ reason, if it is mutated, it will not able to activate its target genes, means that the target gene means like P21 will not be there. Nobody is there to stop the CDK or cyclins. So cyclins and CDK will be unregulated, will be higher in amount, and then it will go. Nobody is there to stop the cells to go for the cell cycle. Cell will proceed without any controls, and that can lead to the tumor. Great. Okay. 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 John का hands free का issue है. Okay, no problem. John, you need to fix these things. Okay. So I think we are done. I don't want you to push you more, but that's okay. Okay. I will ask about this slide. Okay, next time. Whenever we meet again, we will talk on